All right, folks, here we are, week 2. Now we got into chemistry, and I anticipate a lot of questions about some of these intricacies of how molecules and atoms work. So let's just start going into some of these questions and the review topics. I have a question right now about polar bonds. So let's look at polar bonds. Would anybody want to tell me in a nutshell what polar means?
Opposite.
Okay, you think it right as being opposite. You know, we've got North Pole, South Pole. You've got things that are at different ends or that are opposite in each other. So when we're talking about polar bonds, we're talking about areas where there's going to be some partial charge somewhere. So to help talk about this, let me pull up that PDF of our lecture slides so we can look at some actual bonds. Now we're talking about covalent bonds here, right? Polar covalent bonds. And in a covalent bond, what's going on with those electrons? They are being what? Anybody remember the difference between ionic bonds and covalent bonds?
You're sharing, covalent bonds share electrons.
Exactly. So in a covalent bond, they are sharing a pair of electrons. You've got two atoms and there's a pair of electrons, and those electrons are being shared between them. So I'm going to get up these covalent bonds here, and you can kind of see that you'd have, here's an oxygen atom in this image and another oxygen atom, and there are two covalent bonds forming between these two oxygen atoms. That's indicated by this line here. That's called a double bond since it's two covalent bonds. They are sharing two pairs of electrons, and that is making their outer shells complete. So this oxygen atom over here now has 1, 2, 3, 4, 5, 6, 7, 8 electrons in its outer shell, and so does this oxygen atom, and that's stabilizing them both by forming these covalent bonds.
Now, sorry.
What?
Sorry, I don't want to interrupt. I just have a quick question on that.
Okay, good. We haven't quite gotten to polar yet, but we're getting there. Yeah.
You're talking about like the number of bonds that it's making. Is it making the same number of bonds as it's missing electrons?
Yeah, it's trying to become a stable atom. The atom is going to react in a way that fills its outer electron shell. So when we move beyond, so you can see for oxygen here, there's this inner shell that's got 2 electrons, and then as we move beyond that, they can have 8, right? And so they're going to look to fill those outer electron shells to stabilize as an atom. And if we look at Chan here, and so I'm in my PDF of the slides. If you don't know where to find it, go to week 2 module, go to lecture material. In the same little page where I posted the lecture videos, at the bottom is the PDF of all of these slides. And that's what I'm looking at right now. So yeah, Adriana, if you look at these atoms here, and this of course is carbon, hydrogen, oxygen, nitrogen, are 4 elements that make up 96% of all living things. This shows you visually how many electrons each of these would need to become stabilized, to fill that outermost shell. So how many does hydrogen need?
1.
1. And that's because it only has that one innermost shell, and it can only hold 2. So hydrogen will form 1 bond, 1 covalent bond. Carbon needs 4. And so it's going to form 4 covalent bonds. And it can do that in a number of ways, right? It could hook up with 4 hydrogens to fill those needs, or 3 nitrogens. So there are different ways that these bonds can be fulfilled. Sorry, not 3. It could form a couple of double bonds with a nitrogen and then get a couple of places from other places like hydrogen and so forth. But yeah, it's going to be filling that shell to stabilize. And that's why these molecules form. And they form with these covalent bonds. Nitrogen needs 3, oxygen needs 2. And because oxygen needs 2, you will get a stable form of oxygen being the oxygen that we breathe, right? And that's oxygen gas. And oxygen gas is just one oxygen atom and another oxygen atom, and they're double bonded together, and they're both stable at that point.
Thank you. Sorry.
Yeah, no, this is great to talk about because a lot of people might be having the same questions. So now the question is, what makes it polar, a bond polar or non-polar? And I'll ask you all first, are these bonds between these two oxygens polar or non-polar?
Non-polar.
They are non-polar. And the reason they are non-polar is because oxygen will share equally with oxygen. But if you have two atoms that don't share the pair of electrons equally, then one of those atoms is going to be holding those electrons closer to it. And that atom is going to be overall more negatively charged than the other one that's not getting the electrons as much of the time. Okay, so if two atoms are sharing a pair of electrons, but they're not sharing them equally, one of those atoms is going to have more of a negative charge, and one of them is going to have more of a positive charge because the electrons have a negative charge. So that's going to set up a polar molecule. And let's look at this slide here. There's a slide that says, are these electrons shared equally? And it has to do with the electronegativity of the atoms themselves. So carbon and hydrogen. Carbon and hydrogen have low electronegativity. And what that means is they don't pull on the electrons very strongly. So they don't have a high affinity for electrons. Oxygen and nitrogen, on the other hand, they do. They have a stronger pull for the electrons. So you're going to get a polar covalent bond when an atom with low electronegativity is bonding to an atom with high electronegativity.
Can I ask a dumb question?
You can ask a question that I'm sure is not a dumb question. Yes, you can ask any question.
So how do you know or determine if it's low or high electronegativity?
Okay, that is a great question. And I could tell you that I just know because I'm going to tell you right now that carbon and hydrogen are low and oxygen and nitrogen are high. But if you look at the periodic table, you can actually sometimes see periodic tables where they're indicating electronegativity, and it's a calculation of the number of how far away from the nucleus the orbitals are and where those electrons are going to be, and how many protons are in the nucleus itself. So how much is there a pull on those electrons from the protons that are in the nucleus? So it's calculated based on the structure of each individual element, the atoms in each element.
So if an element, the electron is closer to the protons, then it would be a low electronegativity.
So let's go back and look and see. I'm not really sure. So I'm not a chemist, so this might not be 100% accurate, but let me just tell you about my interpretation of it right now. Let's look at carbon and hydrogen and oxygen on our periodic table over here. Sorry, carbon, nitrogen, and oxygen. In each of these, you have those two electron shells. So the outermost electrons are, roughly in the same distance from the nucleus. But as you move over to the right in the periodic table, these elements have more protons in their nucleus. So the more protons will I think overall it's just going to cause more of an attraction there for the electrons. That is my interpretation of it.
Okay, we don't need to cover it. I just
No, and I'm sorry that that's not a satisfactory chemistry explanation for it, but it has to do with the overall structure of the molecule. And so, yeah, just know that relative to each other, and this is going to be relevant to our bonds here, these towards this end, here's the hydrogen, and even carbon's a little bit more towards this end. Over here, they have a lower electronegativity, and oxygen and nitrogen have higher. So the molecule that we're really the most interested in right now regarding polar bonds is which molecule? Which molecule?
Water.
Water
H2O,
molecule?
water, yeah.
Right, water is polar. So we've got oxygen over here with a high electronegativity, and it's bonding with hydrogen over here that has a low electronegativity. And you get this bond that's forming. And most of the electrons, these two electrons that are being shared, they're actually going to be closer to that oxygen end most of the time. They're moving around. So they're not just like staying there, but they're moving around, but they're mostly going to be closer to that oxygen end, which is going to leave you with, if you didn't do this during the lecture, you should do it right now. This molecule of water where you've got oxygen and the two hydrogens on the end, this area down here is partially negatively charged where the oxygen is, and the hydrogen parts are partially positively charged. So they're like weak magnets. They're not like ions that have an absolute charge. They have a general tendency to be negative at the oxygen part of the molecule and positive at the hydrogen parts of the molecule. And that's a polar bond. It's a bond in which the electrons are not shared equally, and it results in a molecule having regions that are partially negative and other regions that are partially positively charged.
Can I ask you in that last slide, like you asked in the lecture, which are the polar bonds? And I just couldn't really, I knew it wasn't the first two, but I was like, I can't really say about these second two.
Right. So how would you determine if they're polar or not? You would see if you have a high electronegative atom bonding with a low electronegativity atom. And if you don't, then they're not polar. So our two low electronegativity are hydrogen and carbon. So if hydrogen is binding with hydrogen, they both have a low pull, so they're sharing equally. It's no problem. Think of a tug of war, right? And you've got two weak participants in a tug of war, and it's not going anywhere. It's equal because they're both weak. With oxygen and oxygen, same idea of a tug of war, but oxygen and oxygen are both strong, still not going anywhere since they're both strong. With oxygen and hydrogen, oxygen is strong and hydrogen is weak. So oxygen's going to pull those electrons more toward its end, and that creates a polar bond. With carbon and hydrogen, like you would have here in methane gas, carbon and hydrogen are both weak. They have low electronegativity. So it's going to be roughly evenly distributed. They're not getting pulled toward one. The electrons are not getting pulled toward one more than the other. So there's not going to be any polarity there. It's going to be non-polar bonds. Now, what if it were nitrogen and hydrogen? Would it be a polar bond between nitrogen and hydrogen? I know some.
Yes,
Yeah.
I think so.
Yeah, it would, right? Because nitrogen has high electronegativity, hydrogen has low. So the nitrogen's going to pull the electrons more, and that nitrogen part of the molecule is going to have more of a negative charge. Does that make sense?
I'm not really sure, but as long as if I just remember these 4 things here, the low and the high, then will I have it covered?
If you remember that carbon and hydrogen have a low electronegativity and oxygen and nitrogen have a high, and you remember that if you bind, if a low electronegative atom is bound to a high electronegative atom, then that's not going to, it's going to be polar. It's not going to be even. So hydrogen and oxygen is polar, carbon and oxygen is polar, hydrogen and nitrogen is polar, carbon and nitrogen, that would be polar because they're not equal. Yeah.
Okay.
Mm-hmm. And there's another question that this gets to. Polarity, I think, is the most challenging concept of these lectures. And once you understand how polarity works and how polar molecules behave, then it's easier to understand how how water behaves, because so much of what we look at about these properties of water has to do with the fact that it's a polar molecule, that the electrons are being pulled more strongly by oxygen, and that leaves the oxygen end of the molecule partially negative, and those hydrogen ends are partially positive, which is why when you get water moving around with other water, it's going to be attracted to other water molecules. It's going to pause in that position where a hydrogen from one molecule is next to an oxygen from the other molecule. And it's just going to kind of be held together that way until it breaks. They do form and unform. But this polarity of water is what leads to these hydrogen bonds forming, right? The hydrogen bonds that are these attractions between 2 different water molecules. And that leads to cohesion and adhesion, surface tension.
Sorry, while you're talking about that,
yeah,
questions for number 6, it says, are these features due to hydrogen bonding or polarity? But if hydrogen bonding is caused by polarity, then I'm like, it's like a chicken egg situation.
I totally agree with you, Adriana. I think you're absolutely right. So let's talk about that question about these properties of water and whether they're due to hydrogen bonds or polarity. And you're right, polarity ultimately does cause hydrogen bonds, but if hydrogen bonds is an option, then you want to go with hydrogen bonds in that case. But there is an example where it's not hydrogen bonds, it's polarity. So let's look at that. Going to these review questions, and that was number 6. Is that what you said?
Yeah.
Okay. So our 4 properties of water that make it well suited to sustaining life. The first one is what?
Cohesion.
Cohesion. Surface tension. Cohesion, you would say, is due to hydrogen bonds. And what that just mainly means is, what is holding the water molecules together? Why are they sticking together? It's because of those hydrogen bonds. It's because of that attraction to each other. So for that one, you would say hydrogen bonds. And you got your little penny there, the hydrogen bonds holding the water together. The ability to resist temperature change. The reason we would absolutely say this is hydrogen bonds is that when you heat up, if you have a skillet of water or a pot of water on the stove, when you're heating that up, the metal pot is going to get hot a lot faster than the water itself does. And that's because as water increases its speed of motion, which is what happens when it gets hot, these hydrogen bonds are breaking more frequently. And when you break a hydrogen bond, it actually absorbs heat energy. And that causes the whole warming up process to be slower for water. So it is those bonds breaking, or in the case of when you're cooling something down, the bonds are forming. And when they form, it releases some heat energy. That is what's going to help regulate that temperature. So yeah, the hydrogen bonds are because of polarity, but it's the hydrogen bonds that are responsible for that slowness to change temperature. And then for this becoming less dense from frozen, it's because of the hydrogen bonds. It's because they get stuck in a certain configuration of spacing between these water molecules when water freezes. So again, it's, water's moving. If it's moving, you can fit more water molecules into this thing. But if there's a hydrogen bond forming, then you have a spreading or a spacing that takes place. And if those hydrogen bonds get fixed into place when the water freezes, then you have a crystal, and it's less dense. So those three were all hydrogen bonds. The only one that you wouldn't say is due to hydrogen bonds is that water is a great solvent. It's a little bit due to hydrogen bonds, but not entirely. And the reason it's not entirely due to hydrogen bonds is that water can dissolve things that are ions like sodium and chloride. It can dissolve anything that has a charge, whether it's a partial charge or a full charge, because water is going to surround and separate things. So if water's dissolving sodium chloride, salt, it is an attraction here. You wouldn't necessarily call it a hydrogen bond, but it's going to cause the water molecules to surround and separate these little sodium and chloride ions in the salt and keep them from forming back into a salt crystal. So does that make sense, Adriana? But I totally get where you're coming from with that.
Yes, that helps a lot. Thank you.
Well, while we're talking about polarity, there's a question I think is challenging. I'd like to bring up, unless anybody else has another question that was related to this. What about this question? We've got our main classes of biomolecules. We were looking at carbohydrates, proteins, lipids, nucleic acids. Which class of biomolecules are mostly hydrophobic? Which ones do not dissolve in water?
Lipids.
It's the lipids, right? The lipids are fats and oils, and they are not soluble in water. So even though water is a fantastic solvent and a lot of things can dissolve in water, lipids do not. So why don't they? What feature of lipids makes them hydrophobic or mostly hydrophobic? Did anybody get an answer for that one? I'm going to move.
I just had a question.
Yeah.
Where are the week 2 review questions? Like, where is that under on Canvas?
Let me show you. Week 2, right? Week 2 module.
Mm-hmm.
And if you're in the week 2 module, it says week 2 review questions.
Oh, thank you. I was like so confused.
And so you just select that, and then it'll give you a link to a Google Doc. And you can't change the Google Doc because then you'd be changing my review questions, but you can make a copy of it and save it as something different and have your own copy there.
Okay, thank you so much.
So I did get a message in the chat that says lipids have many non-polar covalent bonds, and that's why they're hydrophobic. And that is essentially it. Yeah, so let's look at the part of lipids that's going to be these non-polar covalent bonds. they often have a hydrocarbon component to them. So when we look at lipids, I'm sorry if I'm making anybody dizzy right now. Scrolling. All right. A big component of lipids is often that they will have a hydrocarbon ring or a hydrocarbon chain. So this is a hydrocarbon chain that you should be seeing right now. And hydrocarbon rings, we saw some of those, I think, when we were looking at some steroids, but sometimes it's a ring structure, sometimes it's a chain structure. I'm going to go back to this hydrocarbon chain. Right here. So look at this whole structure. Are any of these bonds polar? It's a carbon to a hydrogen, a carbon to a hydrogen, a carbon to a carbon, a carbon to a carbon, a carbon to a carbon. What we call a hydrocarbon is called a hydrocarbon because it's just hydrogen and carbon. And so you've got these long chains of just hydrogen and carbon, which, by the way, is what oil is made of and natural gas is made of, tar. It's hydrocarbons. If you burn them, you break all these bonds. It releases a lot of energy. You get a lot of our energy that we use that way. But these bonds themselves, are they polar? Are any of these bonds polar?
No.
No, there's no polarity here. And remember,
Since both of those are really low in electronegativity,
yeah,
is that contributing to that non-polar aspect?
that's exactly why they're nonpolar, because
Okay.
they're sharing electrons. They're sharing electrons pretty equally. So those electrons are just hanging out in the middle roughly, and you don't have any areas that have partial negative charge and partial positive charge. So
Okay, that makes sense.
because they both have low electronegativity, that they share equally. And there's no partial charge here. This structure does not act like a magnet at all. So water cannot work its magic on this. It has nothing to be attracted to, right? Water is attracted to other polar molecules, or it's attracted to ions like sodium ions and chloride ions, but it can't be attracted to this hydrocarbon because there's no polarity here. It's like wood. It's like a piece of wood in an electric field, right? It's not going to conduct a signal because it's not charged at all. So that's the problem with these lipids and water. Now, sometimes there are polar ends, and we're going to talk a lot about phospholipids in this class. And a phospholipid is a molecule that has a little part that's polar at the top, and most of it's non-polar, these long chains. And you'll see that a lot. Like even when you look at these fats, like a triglyceride, there's a little bit of polarity up here. Anytime you're throwing oxygen into the mix, it's got a bigger tug. So there's going to be a little zone of polarity, but then you have this whole big chain that's all hydrocarbons. And that is not going to let water be attracted to it because there's no charge. And that's why lipids are not soluble in water. Which is why when you get tar on your feet at the beach, you cannot clean that tar off with water. You're going to have to get an oil like olive oil, baby oil, some kind of oil-based substance to get the tar off, because water doesn't work its magic, because the oil is a hydrocarbon, it's a lipid, and it doesn't have any partial charge in those areas. So
I have a quick question.
Mhm.
So for when it says what feature of these molecules make them, would it be their like a non-polar hydrocarbon is what makes them?
yeah, the hydrocarbon chains or rings that are
Okay.
not. Yep.
Thank you.
I have a little drawing of it. I discovered I had a drawing here, right? Yep, this is like a hydrocarbon ring. Non-polar. It's non-soluble in water. And we call that hydrophobic. The word hydrophobic is a little bit misleading because it conjures up images of things being scared of water. But what it really means is that water cannot act as a solvent to dissolve those things. So it's not attracted to water. Okay, any other questions?
I have one. Sorry, I feel like I'm talking too much.
Not at all.
On one of the slides from when we were talking about covalent bonds, it says a molecule is like something with covalent bonds, but then what do we call something that has ionic bonds?
Oh, okay. So the question is, yeah, you're right. A molecule is something that has covalent bonds. So that's how we would define that. An ionic bond could form, like sodium chloride, for example, we would call that a compound. So sodium chloride is a compound, it's not a molecule.
Thank
And
you.
it's not a molecule is because it doesn't have a covalent bond. You can call technically molecule is more specific than a compound too. You can sometimes call things that do have covalent bonds compounds, but you wouldn't call something that's bound together by an ionic bond a compound. Now, another difference between molecules and compounds is that you're not going to have a compound that is the same element. So O2, oxygen gas, would not be considered, that can't be a compound because it's the same element. These are things that you really don't need to focus on, but I appreciate why you would want that clarification. But I'm not going to get into the nitty gritties of what's a compound and what's a molecule and stuff like that. But that is basically the fundamental, is that in order to be a molecule, it's held together by covalent bonds, and they don't have to be different types of atoms. You can have oxygen, oxygen, which is O2, or you can have hydrogen, hydrogen, which is H2, and it's a molecule.
Thank you.
You're welcome. Gonna go back and look at this. How are we doing on protein structures?
For number 15, could you maybe go over like the last part of the question, the what type of bonds form those?
Yeah,
Thank
definitely.
you.
And I think for this one, I probably will do a little drawing too. And see if I get some blank paper here. I'm just gonna walk us through this and give you a different view on it, because I know you have the slides that you can look at. Proteins. Can you just see me right now? Or do you see everybody?
I see everybody.
Okay, I'm gonna share my screen again. And I'll just put this up since you can't see me just slowly. So basically what you're gonna have is, what's the primary structure of a protein? How would you describe that? It's a chain of what?
Amino acids.
Chain of amino acids. Good. The amino acids are the monomers, and they're all going to be linked up, and that's going to produce a polymer, a polypeptide, the chain of amino acids. That's the primary structure. I've got my little toys here. you could think, oh, here's one amino acid. This is phenylalanine. Okay, this is leucine. This is isoleucine. And all of these amino acids are going to go together and make a chain. And that's the primary structure. Then, because of polarity, because this amino acid might have a charge right here, and this amino acid could have a charge, this is going to start, it's going to start folding around and over itself. I can't do it with these toys, but you've got this chain that looks like this, right? Just one chain, but it's going to start bending or folding in a certain way. So the secondary structure is a predictable folding pattern that forms these things called alpha helices or beta pleated sheets. So that strand of protein that you had will start folding on itself. And sometimes it's going to create a helix. And the reason it does that is because polar areas are attracted to other polar areas, and those are hydrogen bonds. So if you have a hydrogen bond forming, it's holding that secondary structure together. So that's answering one of those questions. What holds the secondary structure together? It's hydrogen bonds. So look at this alpha helix right here. This is your protein chain. And this area of the chain is attracted to this other area of the chain. And they're doing so at these regular intervals that's going to produce a certain form like an alpha helix. Or in this case, you've got these two chains that are attracted to each other. These little dot, 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 those are hydrogen bonds. That's going to pull them together and hold them in that secondary structure. Now, do you think, is that going to be a very really stable structure, or could you break those hydrogen bonds?
You can break them?
Yeah, actually, because so much of this protein structure is hinging on hydrogen bonds and these attractions, it's kind of easy to break the protein structure, and that would be called denaturing a protein. And we're going to talk about that in coming weeks, that if you add acid, or even if you make it more basic, or if you add heat, these things could get in there and disrupt this secondary structure and then the tertiary structure, because it's going to keep folding more. So you have these little regions that are alpha helices or beta pleated sheets. But then on top of that, sometimes you're going to get attractions from other regions. So you've got your protein, you might have an alpha helix, and then fold, 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 beta pleated sheet, fold, 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 alpha helix. So there are the secondary structures, and then there's all this other complex folding that occurs, and that complex folding is the tertiary structure of the protein. And that again is because of attractions from different areas. And when it gets into its overall folding pattern, then you have, sometimes you have the protein as it needs to be at that point. But if there are multiple subunits of the protein, like hemoglobin has 4 subunits, then those subunits are going to join together, and that gives you the completed protein itself. So those questions on 15, the secondary structure is the alpha helix and the beta pleated sheets. And the types of bonds are what?
Hydrogen bonds.
Good. Hydrogen bonds. And then what about this question? What is the relationship between the primary structure and DNA? So that's asking you about the relationship between that chain of amino acids and DNA. I'm going to go back over here. I'm just going to look at this chain of amino acids. Glucine, serine, lysine, cysteine, these are all the amino acids. Does the order of these amino acids matter? Do you think it's important to have these amino acids in this specific order to make this protein?
I would guess yes, because like if they're in a different order, then the polarity is in different spots.
Exactly, right? If you don't get this chain of amino acids with the right amino acids, then that's going to upset everything, right? It won't fold correctly. So you can think of this amino acid chain as being like a word that has letters in it. And you've got to get the letters in the word correct so that that word can be interpreted. Well, how does a cell know what amino acids to put in this chain? In a cell, you're going to build this protein. So this amino acid serine, and then we're going to have lysine and cysteine and proline. And don't worry about those names right now, or really ever in this class. But how does the cell know which order to put these in?
It is determined by the DNA.
That's what our DNA does. Yeah. So, I mean, that kind of the fundamental job of DNA is to have a code, these A's, G's, and T's and C's, and that code that's in the DNA tells the cell which proteins to make and how to make them. It codes for the sequence of amino acids. So if you have a mutation in your DNA and that DNA is part of a gene that codes for a specific protein, then what can happen is you can end up with the wrong amino acid sequence, and that leads to the formation of a protein that's the wrong shape that doesn't do its job. So it's really fundamentally important, the structure of proteins, and it's our DNA that codes for the production of these. And what that means is everything that we know about how important our DNA is, it's only affecting us through our production of proteins. So that gives you an idea of how important these proteins are, right? Any mutation that you have that might affect something about you, it's affecting you because you're not making the right protein for that thing anymore. So, all right, proteins do a lot of things.
Can I ask a kind of off-topic question about protein?
Maybe.
When people for fitness stuff, when people are trying to recomp, they eat a lot of protein, but you said protein isn't energy. Like, why is eating protein beneficial to muscles?
Because your muscles are made of protein, and to build proteins, what do you need?
Amino acids.
You need amino acids. So some amino acids we can make, some amino acids we have to get from the food that we eat. But if you're trying to build a lot of muscle cells, build a lot of muscle tissue in your body, then it's useful to have easy access to amino acids. You're not going to be using the proteins that you're eating, but you could use the amino acids from those proteins to build proteins within your body.
Okay, that makes sense. Thank you.
All right, and I think if there aren't any other questions, I'm going to leave you all with this this idea, which is we talked about the functions of these different classes of biomolecules, and I think of all of those, it's pretty straightforward what carbohydrates do, what nucleic acids do. Lipids have various functions. But proteins, really, man, these proteins can do so many things in our bodies. They can be enzymes. Every single reaction that we have in our body is catalyzed by a specific enzyme, which is a specific protein. They provide structure. They do transport. They are antibodies that we use in fighting off diseases. They are controlling what goes in and out of cells. The list just goes on and on. If it comes up on an exam, I would want you to know these specific functions of proteins. What we would not say that a function of proteins is to provide energy for our bodies, because generally if we need energy, we're going to get energy from carbohydrates and from fats. But proteins as biomolecules, as molecules inside our body, those are not things that are providing energy for us. They are things that are carrying out these specific tasks. And those are the proteins that we are making in our bodies. So just like Adriana just said, we need to eat proteins for various reasons, and they also kind of help us feel full longer. We get amino acids that way, and so forth. Okay. Well,
Thank you.
that
Thank
was
you.
I think that was pretty good, getting on most of our main points of what I thought were some of the more challenging concepts here. Any more questions before we go? All right. Well, I hope I get to see you next week. If you are in my lab section and you're going to come to our wrap up, then it is a different link. I'm going to actually try to change that in the future, so it's the same link. You don't have to leave and come back. But it will be a different link, and it is the lab wrap up link for my sections that you can find at your Canvas site. So, all right.
Quick question.
Yeah.
If my lab teacher is the other teacher, would I be able to go on to yours one day if I had like a question about a lab?
Yes.
Okay.
Absolutely. Yeah.
Okay, great.
And the same would apply to students who are in my lab sections. If you want to show up to one of Liz's sessions, then, I mean, if it's a specific question about how your lab was graded, then the other instructor is not going to be able to answer it. But if it's a general question, we would love to have you.
Okay, great. Thank you so much.
Okay, thanks, y'all.
Thank you.
I actually have one question. It's
Thank
not
you.
about like the labs or anything. On the next Zoom for the wrap one, you think you can show how to use like the